Welcome to Lab Unit 3. In this unit, we are going to be focusing on unknown organisms that are all gram-negative bacilli. So with this unit, you are going to want to focus on observations, results, and interpretations as they apply to the different media and tests that we are going to talk about as we seek to identify our 8 unknown organisms. And the 8 unknown organisms that you will be studying are listed on this slide. They are covered by the captions, but you have them on your PowerPoint. And as you can see in the picture, if you were to do a gram stain, you would see red rod-shaped cells all in a single arrangement. Okay, so that's what they're all going to have in common. So we're going to talk about various selective and differential media that we can use to identify each individual organism. Another thing that all of these unknown organisms have in common is that they are enterics. So enterics are bacteria that are going to be found and associated with the GI tract. So some of these organisms are going to be part of our normal microbiota, in which case several of those are going to be opportunistic pathogens, meaning if they have the chance to become pathogenic, they'll take it. But normally they're just there in our digestive system, doing their thing. You know, maybe they help with digestion, help fight off pathogens. But again, they're opportunistic when given the opportunity. Some of them are just strictly pathogens, and so they are introduced to our digestive tract via consuming contaminated food or water. There are 3 pathogens that we're going to be learning about, and I'm not going to go through each of these next 3 slides because we've already talked about these pathogens and the diseases in the lecture. Okay, so this is just kind of a nice recap of these organisms and their associated disease. So Salmonella enterica can cause salmonellosis. Salmonella typhi can cause typhoid fever. And lastly, Shigella flexneri causes shigellosis or bacillary dysentery, which is more commonly known as bloody diarrhea. So in this unit, again, please make sure you're paying attention to, is the media selective and or differential? And then what are the observations associated with that media? So what would you physically see when looking at the plate or the broth or the slant? What are the results? Oftentimes that's positive or negative. And then your interpretation is always the so what. What does it mean that you observed hot pink colonies? What does it mean that it's a positive result? So with your interpretations, you're always going to go back and look at the purpose of the media. Why did we streak our organism on a McConkey plate? What are we hoping to learn about the organism? Okay, so that's going to always go back to what your interpretation is. All right, y'all, in the remaining videos, we are going to go through the various media used to identify our gram-negative bacilli. Let me know if you have any questions.